Right now, like as you are listening to this very sentence, there are millions of chemical reactions happening inside your body.
Millions, at least.
Literally millions. And it is not some, chaotic microscopic soup in there. It's perfectly choreographed.
Right, it's highly organized.
It's like the most complex high-speed ballet imaginable happening in every single one of your cells.
Yeah.
But how does your body actually pull that off?
That is the big question.
Exactly. So today, we are taking a deep dive into a really comprehensive stack of biological texts that map out the ultimate instruction manual of life, which is your DNA, and the microscopic molecular machines that code builds, called enzymes.
It's a remarkable journey through biology, honestly. The sources we're unpacking today cover everything from the structural architecture of proteins to the, incredibly precise mechanics of gene expression.
Yeah, it gets really detailed.
It does. We are essentially going to trace how the macroscopic world, like, the food you eat for breakfast, connects directly down to the atomic level of how your cells function and replicate and just survive.
Okay, so let's unpack this. Because to understand the code of life, the DNA vault, we first have to understand the workers that the code is busy building, the enzymes.
Right, the workers on the factory floor.
Yeah, and while I was reading through the source material, I kept thinking about this section called Thinking Bigger, which analyzes some very popular claims from healthy eating blogs.
Oh, the raw food diet stuff.
Yes, there are authors out there heavily promoting raw food diets, and their central argument revolves around enzymes.
Right.
They claim that when you cook food, the heat destroys the food's natural enzymes. And because those plant enzymes are, dead, the theory goes that your own digestive system has to overwork itself to make new enzymes from scratch to digest the meal.
Which eventually exhausts your organs, according to the blog.
Yeah.
Yeah, that narrative has really deep roots in wellness circles. The underlying assumption is that you need to consume intact, quote-unquote, living enzymes from your food to stay healthy and preserve your body's energy.
Which, I have to be honest, it kind of made me laugh once I read the actual biology.
It's a bit of a stretch.
Because to me, that sounds like saying, hey, my car's transmission is broken, but if I just eat a mechanic, I'll suddenly have the functioning tools inside my stomach to fix my car.
That is a very visceral analogy, but it's accurate.
Right. Do we actually need to eat intact functioning enzymes from our food to run our digestion?
The short answer is no, not at all. But to see why that blog claim is biologically flawed, we need to look at what an enzyme actually is and, what it does.
Okay, lay it on me.
So the sources define enzymes as globular proteins. Their primary job is to act as biological catalysts.
Meaning they speed things up.
Exactly.
But how do they actually do that? Because the text mentions this term, activation energy.
Right. Think of activation energy like a steep hill that a boulder has to roll over before it can, gain momentum and roll down the other side.
Okay, I'm picturing it.
For any chemical reaction to happen in your body, it has to get over that initial energy hill. Without enzymes, the reactions that keep you alive would happen far too slowly at your normal human body temperature.
So you just die.
You simply wouldn't survive, yeah. Enzymes solve this by essentially tunneling through the hill.
Oh, wow.
They lower that activation energy, allowing these vital reactions to happen rapidly and efficiently. Now, the raw food bloggers aren't completely wrong about one thing. Heat does heavily impact how these enzymes operate.
I noticed that. The sources break down the math of it with something called the temperature coefficient, or the Q10 rule.
Yes, Q10.
And this Q10 curve is exactly why those raw food advocates are so obsessed with heat. The rule states that between 0 degrees and 40 degrees Celsius, the rate of an enzyme-controlled reaction actually doubles for every 10-degree rise in temperature.
And the mechanism behind that doubling is purely kinetic.
Like movement.
Exactly. As you add heat, the molecules vibrate faster and move around more. They collide with each other more frequently and with way more energy, meaning the enzyme and its target molecule find each other faster.
So the reaction rate just ramps up dramatically.
It does.
Until it hits that crucial 40 degrees Celsius mark.
Yeah.
And then crash. The protein loses its shape and the reaction stops almost completely.
Right. The text calls this denaturation.
But what is physically breaking there?
So the three-dimensional shape of the enzyme is actually unraveling. Enzymes have a very specific area called an active site.
The lock and key thing.
Well, historically, scientists use a lock and key model to describe this, suggesting the target molecule, the substrate, fit rigidly, perfectly into the active site.
Like a metal key in a padlock.
Right. But the modern, more accurate model from our sources is the induced-fit hypothesis.
The induced fit. So it's not a rigid metal lock. Is it more like a flexible glove molding to a hand?
That is a perfect way to visualize it. As the substrate enters, the active site actually changes shape slightly to wrap around it. It forces the reaction to happen, releases the new product, and then springs back to its original shape.
That's incredibly dynamic.
It is, but those intricate shapes are held together by relatively weak internal bonds. When the temperature pushes past 40 degrees Celsius, the kinetic vibration becomes so violent that it snaps those weak bonds.
Oh, I see.
The glove melts and deforms permanently. The enzyme is denatured.
So if the glove melts, the bloggers think we're doomed. They think we've lost the workers.
Right.
But that entire argument falls apart because of what our digestive tract actually does to food. The source material points out that our stomach and intestines, they don't absorb whole proteins anyway. We break them down.
We completely dismantle them.
Yeah, we actively chop those long, complex protein chains into their tiny basic building blocks, the amino acids.
And that complete dismantling is why the raw food argument completely misses the mark. Whether the enzyme in that broccoli you ate was alive and perfectly folded or, denatured and melted by boiling water, your digestive acid and your own enzymes are going to completely shred it into basic amino acids regardless.
You absorb those raw materials.
Exactly. You absorb them, and your cells use them to manufacture your specific human enzymes. You never rely on a plant's enzymes to run your metabolism.
You don't need to eat the mechanic. You just break down the mechanic into spare parts.
Right.
And use them to build your own tools.
Mm.
And the fact that our bodies just recycle these amino acid parts answers the diet question perfectly.
Mm-hmm.
But what really struck me in the text is that it took the scientific community decades to figure out that enzymes were just regular proteins made of these parts.
Oh, yeah. The history there is wild.
There's a story in here about a scientist named James B. Sumner.
Sumner's story is a fascinating piece of scientific history. In 1926, he achieved something literally unprecedented. He managed to extract the very first pure crystalline enzyme.
It was urease, right, from jack beans?
Yes, urease from jack beans. And his analysis proved definitively that this enzyme was a protein.
But nobody believed him.
No, they didn't.
The established scientists just completely dismissed his work.
They rejected it entirely. At the time, enzymes were somewhat mysterious, almost thought of as magical, vital forces. Or substances too complex to just be standard proteins.
Wow.
Established researchers had been trying and failing to isolate them for years. It took a full 20 years for the scientific consensus to catch up to Sumner's data.
20 years.
Yeah, he finally won the Nobel Prize in 1946 for proving that these vital biological machines were just intricately folded protein chains.
Imagine that. 20 years of holding the fundamental secret of biology, just waiting for the world to realize you were right.
It's incredible patience.
So if our bodies are just building their own perfectly shaped protein gloves from scratch using recycled amino acids,
Yeah.
where do the blueprints come from? Like, how does a cell know how to assemble these parts into a specific enzyme? Which leads us to the vault, the DNA.
The ultimate instruction manual. It dictates the design of every single enzyme your body produces.
Now, everyone recognizes the famous shape of DNA, the double helix, but the text gets heavily into the chemistry of it, calling DNA a polynucleotide.
Right.
It lists all these components: a 5-carbon pentose sugar called deoxyribose, a phosphate group, a nitrogen-containing base. It feels very dense. What does this actually look like in practice?
Think of a polynucleotide like a long chain link fence that's been twisted into a spiral. The structure has a very rigid backbone. The deoxyribose sugar and the phosphate group alternate to form the strong outer rails of this fence.
Okay.
They are locked together by what the text calls phosphodester bonds. You can think of those bonds as the heavy-duty welds holding the metal frame together.
Okay, so the sugar and phosphate are the heavy metal frame. Where is the actual information? Where is the code?
The code is in the middle. Sticking out from that sugar-phosphate rail are the nitrogen-containing bases. These are the rungs of the ladder or the connectors linking the two sides of the fence together. And there are strict chemical rules for how these bases pair up across the double helix.
Right. The text breaks these down into purines and pyrimidines: adenine, thymine, cytosine, and guanine. The classic A, T, C, and G.
And their pairing is non-negotiable. A purine must always pair with a pyrimidine to keep the width of the helix perfectly uniform.
Makes sense structurally.
Specifically, adenine always pairs with thymine, held together by two weak hydrogen bonds, and cytosine always pairs with guanine, held by 3 hydrogen bonds.
A pairs with T, C pairs with G, always.
Always.
But this strict pairing rule brings up something I was genuinely confused about while reading. The sources discuss how scientists used to argue about how this double helix copies itself when a cell divides.
Ah, yes. The replication problem.
Yeah, I was reading about the conservative model where the original DNA supposedly stays completely intact and a brand new double helix is just built next to it. But chemically, I don't understand how a closed, twisted fence could somehow force an exact copy to be built outside of it without opening up.
Many scientists shared your exact confusion. The alternative theory was the semi-conservative model, which suggested the helix unzips down the middle, breaking those weak hydrogen bonds between the bases. Right. Then each single half serves as an exposed template to build a new partner.
Which makes logical sense, but how do you actually prove that? You can't just, put a tiny camera inside a cell.
No, you can't.
So the sources detail this experiment by Matthew Meselson and Franklin Stahl in the late 1950s using E. coli bacteria. And it is incredibly clever.
It is widely considered one of the most elegant experiments in biology.
Yeah.
Meselson and Stahl grew E. coli in a broth where the only source of nitrogen was a heavy radioactive isotope called nitrogen-15.
So as the bacteria reproduce, they pull that heavy nitrogen from the broth to build those nitrogen bases in their DNA.
Exactly.
Meaning their entire DNA structure is physically heavier than normal DNA.
Right. Then they took those heavy bacteria and transferred them to a new broth. But this broth only contained regular light nitrogen, nitrogen-14. They allowed the bacteria to replicate their DNA exactly one single generation. Then they extracted the DNA and spun it in an ultra-high-speed centrifuge.
Now, I understand the spinning, but how does a centrifuge actually show us whether the DNA unzipped or stayed intact?
Well, spinning at those extreme speeds creates a density gradient in the tube. Heavy molecules sink toward the bottom and lighter molecules float near the top.
Oh, I see.
If the conservative model was true, if the original heavy DNA stayed completely closed and intact and just spit out a brand new copy made from the light nitrogen in the new broth, you would expect to see 2 distinct bands in the tube.
One heavy band at the bottom from the parents.
Yep.
And one light band at the top for the new copy.
Exactly, but they didn't see 2 bands.
They found exactly one single band.
One band.
And its weight was perfectly dead center, halfway between heavy and light. It was a complete hybrid.
Which was the visual proof needed for the semi-conservative model. The original heavy double helix unzipped down the middle. Each heavy strand then acted as an exposed template, and new light nucleotides from the environment paired up to it. A to T, C to G.
So every new DNA molecule in that bacteria was exactly half old and half new.
Half heavy metal, half light metal. It's an undeniable result.
And to actually execute this unzipping and rebuilding process, the DNA relies on, drumroll please, enzymes.
It all comes back to enzymes.
The sources mention 3 amendments. You've got DNA helicase, which acts as the actual zipper pull, breaking those hydrogen bonds and unwinding the strands.
Right.
Then DNA polymerase comes in and lines up all the new matching nucleotides along the template. And finally, DNA ligase acts like the welder, locking the new sugar phosphate backbone together.
The profound part of that mechanism is the biological loop it creates. The cell requires these specific enzymes to replicate the DNA, but the DNA vault contains the very instructions required to build those specific enzymes in the first place.
It hurts my brain in the best way possible. But wait, this brings up a huge logistical math problem.
Oh, the translation issue.
Yes. We established that the DNA code only uses 4 letters, A, T, C, and G. But proteins, the enzymes, are built from 20 different kinds of amino acids.
Correct.
How do you write 20 unique instructions using an alphabet of only 4 letters?
The math forces an ingenious solution, known as the triplet code, because think about it, if the cell read the DNA 1 base letter at a time, you could only ever code for 4 amino acids.
Right. And if the cell read the letters in pairs, you'd have 4 times 4, which is 16 possible combinations.
Which is still short.
That is still short of the 20 amino acids we need to build our biological machines. So nature had to use groups of 3, a triplet code, which gives you 4 times 4 times 4, 64 possible combinations.
Exactly. 3 bases on the DNA code for 1 single amino acid. This 3-letter sequence is called a codon.
But because there are 64 possible codons, and we only need to code for 20 amino acids, the text uses a very specific word to describe the genetic code. It calls it degenerate.
Yes, a degenerate code.
Now, usually when we call something degenerate in everyday language, it implies it's falling apart, or it's flawed, or losing its function. Why on earth is a degenerate code a positive thing for our survival?
In the context of genetics, degenerate simply means redundant. Because we have 64 combinations for only 20 amino acids, almost every amino acid is coded for by multiple different triplets.
Okay, give me an example.
Think of it like having 4 different toll-free phone numbers that all route to the exact same customer service desk. For instance, the amino acid valine can be summoned by the DNA sequence CAA, CAG, CAT, or CAC.
Oh, so if one number goes down, the message still gets through.
Precisely. Let's say a point mutation occurs, a tiny chemical error where one single letter in the billions of DNA bases is accidentally swapped out during the copying process.
Right.
If your DNA is supposed to read CAA, but the last letter mutates to a G, making it CAG,
It still codes for valine.
the mutation occurs, but the final protein doesn't change at all. The enzyme still folds perfectly, the active site remains perfectly shaped, and you continue to digest your food and run your metabolism normally.
That is wild.
The degenerate nature of the code is a built-in shock absorber. It protects organisms from the potentially catastrophic effects of random genetic mutations.
That is incredibly smart engineering. Okay, so we have the blueprint. The DNA vault is safe. It copies perfectly using the semi-conservative zipper method, and the triplet code has this redundant failsafe.
Ooh, looking good.
But we still have a massive location problem.
The physical separation between the vault and the factory.
Yes. The DNA molecule is massive. It's physically trapped inside the nucleus of the cell. It cannot leave.
It's too big.
But the factories that actually build the proteins, the ribosomes, are way out in the cytoplasm. So how does the recipe get out of the vault? It's like having a master recipe book in a highly secure library. You are absolutely not allowed to check the book out. You can't take it to your kitchen.
No, the master copy stays put.
So you have to go into the library, transcribe the exact recipe you need onto a piece of scratch paper, and take that temporary paper out to your kitchen.
That analogy maps perfectly to the process. And that molecular scratch paper is called RNA, specifically messenger RNA or mRNA.
Okay.
This entirely separates the safe storage of the master code from the chaotic environment of the factory floor.
So let's walk through this manufacturing process. How do we get from the vault to the finished protein?
It happens in 2 main stages, transcription and translation. Transcription happens inside the nucleus. A section of the DNA, just the specific gene needed, unwinds.
Unzips.
Right. An enzyme called RNA polymerase builds the mRNA strand along the exposed DNA template.
But the mRNA isn't an exact carbon copy, is it? The sources point out some key chemical differences. It's single-stranded, so it can slip through the pores of the nuclear vault.
Correct.
It uses a slightly different sugar, and crucially, it doesn't use the letter thymine. The text says it replaces thymine with uracil.
Yep, the U.
Why reinvent a completely new letter? Why not just copy the T?
The use of uracil acts as a biological tag that marks the RNA molecule as a temporary copy rather than permanent storage.
Oh, that makes sense.
It distinguishes the scratch paper from the master blueprint, ensuring the cellular machinery treats them differently. So during transcription, if the DNA template reads adenine, the mRNA scratch paper pairs it with a uracil.
Okay, so this single-stranded, uracil-stamped mRNA detaches. The master DNA zips back up safely in the vault, and the mRNA travels out into the cytoplasm to find a ribosome factory. This kicks off the second stage, translation.
Exactly. The ribosome binds to the mRNA tape. Now, the cell needs to bring the correct amino acids to the factory in the exact order the mRNA dictates.
Right.
This heavy lifting is done by a third type of molecule, transfer RNA, or tRNA.
Now, the text describes tRNA as having a really distinct shape. It folds back on itself to look like a tiny clover leaf.
Yes, a clover leaf.
And on one end of the clover, it carries a specific amino acid. On the other end, it has a sequence of 3 exposed bases called an anticodon. I'm trying to picture the mechanics here. If the mRNA is a long strip of tape with a 3-letter code, how does the clover leaf know where to dock?
Think of the anticodon like a barcode scanner that only recognizes one specific product.
Yep.
If the mRNA codon on the tape reads AUG, a tRNA clover leaf floating in the cytoplasm with the perfect complementary anticodon UAC will dock perfectly onto that spot.
Right.
And that specific tRNA will be carrying the amino acid methionine. The ribosome locks it in place. Then the ribosome ratchets forward to the next triplet codon.
Another matching tRNA docks with the next amino acid.
And an enzyme immediately forms a strong peptide bond between those 2 amino acids.
It's literally an assembly line. Reading the tape, dropping the part,
Yeah.
welding it together, reading the tape, dropping the part.
Exactly like an assembly line.
And the empty tRNAs just float away to grab more raw materials. This chain grows until it hits a specific stop codon that tells the factory the enzyme is finished. And what blows my mind is the sheer scale of this. The source mentions something called polysomes. It's not just one ribosome reading the scratch paper.
Oh, not at all. You can have whole groups of ribosomes, polysomes, trailing along the exact same mRNA thread, one right after the other. It is high-volume, continuous manufacturing.
Going back to the opening thought about the millions of reactions happening right now, your body needs a constant, massive supply of these specific enzymes just to maintain homeostasis. So what does this all mean for you, the listener? Let's connect the dots.
Let's do it.
You eat a meal. Your digestive system dismantles those proteins down into basic amino acid spare parts. Those raw materials are absorbed and sent out to the factory floor of your cells. Meanwhile, deep in the vault of your nucleus, a highly secure, redundant code zips open, prints a temporary messenger copy, and sends it out. And your cellular factories use that message to reassemble those digested raw materials into the exact, perfect, flexible fit enzymes you need to stay alive. It is a breathtaking system of recycling and manufacturing.
It truly is one of nature's greatest marvels. It really is. But if we want to leave you with something that will fundamentally shift how you view your own biology, there is one final detail in the source material that is absolutely staggering.
Lay it on me.
We've spent this whole time discussing genes, the sequences of DNA that code for proteins and enzymes. But according to the text, in a human being, those protein coding genes make up only about 2% of your entire DNA.
Wait, what? Only 2%?
Yes. The other 98% of the human genome is what they call non-coding DNA. It doesn't contain recipes for proteins at all.
That is absurd. You're telling me that 98% of the vault is just empty space. Is it just like evolutionary junk?
That is exactly what scientists called it for decades, junk DNA. But they are now realizing how deeply wrong that assumption was. That vast, unseen 98% is heavily involved in complex regulation.
Regulation.
Yeah, it acts as the management system of the factory. It controls when certain genes turn on, when they turn off in response to your environment, and how different genes interact with each other across the genome.
So they aren't the dancers in the ballet, they are the choreographers.
A perfect way to look at it. But the thrilling truth is, for a huge portion of those non-coding sequences, the scientific community still doesn't know exactly what they are doing.
Okay, that gives me chills. Think about that. Every perfectly choreographed chemical reaction keeping you alive right now is driven by microscopic workers, built by a code. But if only 2% of your DNA actually builds the machines that run you, and the other 98% is complex, mysterious management, what undiscovered instructions, what ancient evolutionary secrets are still quietly sitting there right now in the dark vault of your cells?
It is a profound mystery and one of the greatest frontiers left in biology.
It really is. Keep wondering, keep learning, and we'll catch you next time.